morning, I am Madhurima Datta, Zoology and Biology educational content creator. Now's content for B.Sc and M.Sc in Zoology Honours, Physiology Honours and Biochemistry Honours and also Botany Honours, and exam for CUET PG exam, GATE exam, DBT exam, IIT JAM exam and CSI NET Life Sciences exam. Paper Immunology, Chapter 2, Cells and Tissue of the Immunosystems, and today I will discuss about the phagocytes. Cells of the immunosystem. The cells that serve specialized role in innate and adaptive immunoresponse are called phagocytes. Dendritic cells and antigen-specific lymphocytes and various other leukocytes that junctions to eliminate antigens. These cells are almost all derived from hematopoietic stem cells in the bone marrow, which differentiate along branching lineages. Based on their common precursors, immunocells are broadly classified as either myeloid cells, which include phagocytes, and most DCS, that is dendritic cells, or lymphoid cells, which includes all lymphocytes. Most helper T cells express a surface protein called CD4, and most cytotoxic T lymphocytes, that is CTLs, expresses a different surface protein called CD8. Next, immunocells, mean number and normal range. First is white blood cells. Mean number is 7,400 and normal range 4,500 to 11,000 per millimeter cube. Next, neutrophils. Mean number 4,400 and normal range 40 to 60%. Next, eosinophils. Mean number 200 and normal range 1% to 4%. Next, basophils. Mean number 40 and normal range less than 1%. Next, lymphocytes, mean number 2500 and normal range 20% to 40%. Monocyte, mean number 300 and normal range 2% to 8%. Next, what is the phagocytes? Phagocytes, including neutrophils and macrophages, are cells whose primary function is to ingest and destroy microbes and remove damaged tissue. Description of the phagocytes. The functional response of phagocytes in host defense consists of sequential steps. First, recruitment of the cells to the sites of infection and recognition of and activation by microbes. Next, ingestion of the microbes by the process of phagocytosis and destruction of ingested microbes. Next, blood neutrophils and monocytes, which differentiate into macrophages after entering tissue, are produced in the bone marrow, circulate in the blood and are recruited to sites of the inflammation. Next, the functions of the phagocytes are important in innate immunity and also in the effector phase of some adaptive immunoresponses. And macrophages in tissue can live for long periods so that the macrophage responses may last for a prolonged time. Macrophage responses rely more on include gene transcription and protein expression. Next cell is neutrophils. What is the neutrophils? Neutrophils are the most abundant population of circulating white blood cells, and the principal cell type is acute inflammatory reactions. Description of the neutrophils. Neutrophils circulate as spherical cells approximately 12 to 15 micrometer in diameter with numerous membranous protections, and the nucleus is segmented into 3 to 5 connected lobules. Next, neutrophils are also called polymorphonuclear leukocytes to contrast them with mononuclear cells, that is macrophages and lymphocytes whose nuclei are not multilobed. And the cytoplasm contains two types of membrane-bound granules. The majority of these granules, called specific granules, are filled with the enzymes such as lysozyme, collagenase and elastase. These granules do not stain strongly with either basic or acidic dyes, that is hematoxylin and eosin respectively, which distinguishes neutrophil from two other types of circulation, that is leukocytes and cytoplasmic granules called basophils and eosinophils. Next, the remainder of the granules of neutrophils called the agerophilic granules because microbicidial substances including defenses and cathelicidins. Neutrophils are produced in the bone marrow and arise from the precursors that also give rise to a circulating monocytes. Productions of neutrophils is stimulated by granulocyte colony stimulation factor, that is GCSF, and granulocyte macrophages E colony stimulation factor, that is GM-CSF. An adult human produces more than 1 into 10 to the power 11 neutrophils per day, each of the which circulates in the blood or a few hours or up to 5 days before dying. Neutrophils may migrate to sites of infections rapidly after the entry of microbes, and after entering new tissue, neutrophils functions are only 1 to 2 days, and most of them then die. Next, neutrophils may secrete granules contains also extrude their nuclear contents, that is forming neutrophil extracellular trap or nets which serve to immobilize and kill extracellular microbes but also may damage the healthy tissue. This is an important question under the NAPE of graduation. It is the short note of neutrophil and marks remaining 5 to 10 marks. Now the topic has ended. Please subscribe my YouTube channel Biogenesis by Madhurima and follow my Facebook account Madhurima Datto.